Well, good evening, Beijing, and good morning, Ann Arbor. I'm so glad that we can gather again, as we do for our joint institute, to discuss issues of mutual interest. Let me start off by just introducing myself. I'm Joe Kolars. I'm Senior Associate Dean for Education and Global Initiatives here at the University of Michigan. But more relevant to this discussion, I'm co-director for the Joint Institute that we've had with Peking University Health Science Center. And I've just been so thrilled to be able to work over the years with President Zhang, with Ning Zhang, and so many colleagues. But today, we're actually going to gather for a very focused discussion on mutual awareness and shared opportunities in cancer. And we've just got a fantastic panel lined up. And shortly, I'll be turning it over to the moderators, Max Wicha and Ning Zhang. But before doing so, if I could just take an opportunity for those of you who may be a little bit new to the JI to say a few things. Next slide. For this conference, just to remind you, we're going to be starting what I hope is a longer conversation on cancer immunology, talking about some strategic themes, but more importantly, getting some ideas going between colleagues here at Ann Arbor and at Peking University, having a panel discussion that will be moderated by Ning and Max, and then just a closing remark. Next slide. The Joint Institute actually got started back in 2010 with the idea that perhaps if we tried to create a platform that brought two great institutions together, we could do more than just having investigators find themselves. One of the hallmarks of our Joint Institute is every fall we gather, alternatively between Ann Arbor and Beijing, and we have usually 100 people travel one way or the other. This is one of our last pictures here before COVID. As you can see, we have our own President Mark Schlissel here and President Zhang. And this keeps gathering more and more momentum because what we have is a platform where investigators can find each other and do things that they couldn't do alone. Great talent can find its way forward. So we should be pursuing projects and ideas that are really codependent on each other. And when you have two great institutions that are looking at problems in different ways with different resources, it's really been a win-win dynamic. Our JI has been deemed extremely successful. In terms of NIH dollars, we're just over $30 million. We have almost 120 joint papers that have been launched. And we have a model of collaboration that I think is the envy of many, many institutions, particularly during this time when people are starting to question how the United States and China can work effectively together on issues of common concern. Who isn't concerned about health? But people have applauded us from many different arenas. And we have the opportunity now to take this forward and to start to focus on a kind of a new area. We've had some projects in cancer and immunology, but this is an area we'd like to prioritize. Next slide. In reflecting on our 10 years of success, we went through a strategic planning process, and our vision is really to be this unique international partnership that can enable top researchers from both of our institutions to advance medicine in the US and China and beyond. And again, how can the sum be more than our parts in terms of we come together in unique ways for creativity? We've decided to advance, and instead of just throwing a net out there and see who wants to collaborate, let's prioritize some scientific themes, define focused research priorities, really want to make sure we're cultivating high quality data sets. Our secret sauce, so to speak, has been JI enabling functions, core lab functions that bring us both together, and to really advance and promote our competitive platform, so that others can see we have access to high quality cohorts, that we could potentially leverage with industry for sustainable financial models. Next slide. So what are those scientific themes and research priorities? Well, at the top of the list is the one that's brought us together today, cancer immunology and inflammation. Huge priority for us, a huge priority, I know, for China. And the burden of disease in both of our countries call on us to be more creative and more collaborative on how we can tackle this together. We also have similar priorities in neuroscience and precision health and artificial intelligence, where we'll also be prioritizing our work. Next slide. But with those few introductory comments, I just couldn't be luckier to have the panel that's been assembled here today. And I would like to turn this over to our moderators. Ning Zhang, just a long-term partner from Peking University. Ning and I have done so much together. As many of you know, Ning has been the Vice President for Research at Peking University Health Science Center, a renowned tumor biologist who actually studied at Johns Hopkins under our president, Mark Schlissel, who you saw in that picture, but has just been an outstanding scientist. And he will be co-moderating with one of our luminaries here, Max Wicha. Max actually founded our first cancer center here. And you can't find anyone in the field, I think, who is published or is referenced more when it comes to cancer stem cells. And Max has also succeeded in having a project through our Joint Institute. And he's intrigued about the possibility of pulling together people in different ways to find out what can we do together that we couldn't do alone. So, so Ning and Max, thank you for co-moderating this session. And I know we have a number of speakers and then a discussion, so I'm going to turn things over to you.
Thank you, Joe. All right, thank you, Joe. Good morning, Michigan, and also good evening to the Beijing partners. It's really a great honor for me to be a moderator for this session. And Joe and I talked a lot in the last several months. And the thing we were talking about is how to improve and how with our successful collaboration between the two universities. So Joe proposed this idea of version 2.0, which is that we have proposed to focus, to be more focused, and also to introduce more collaboration and panel discussion. And today will be our first discussion. And in the Beijing side, we will have 3 speakers. I would like to have the honor to introduce our first speaker, Professor Qiming Zhang. Professor Zhang is the academician of Chinese Academy of Engineering, and he also served as a vice chairman of the University Council and the director at the Peking University International Cancer Institute. Dr. Zhang is also the engineer of the China biomedical development, and he participated and organized many strategic planning for the country. Professor Zhang's research focused on the molecular pathways involved in the control of cell cycle checkpoint and apoptosis of DNA damage, and the signal pathways involved in the regulation of the maintenance of genome stability and the tumor metastasis. And in recent years, Professor Zhang paid more attention to the translational study, and also including the genomic study as well, and also the study involving large data set, health data set. And his research has successfully attracted multiple grants from different national agencies. And Dr. Zhang has published more than 240 peer-reviewed papers, including Nature, Cell, etc. And many of these publications have actually helped us to understand more about cancer and also enlighten us on the translational study. So without further ado, Dr. Zhang, please.
Okay, thank you, Ning, for your nice introduction. So my dear colleagues from both Michigan and Beijing, during the introduction delivered by Joe, and the one picture actually we had the last time during our visit, the PKU delegation visit the Ann Arbor. So it actually brought me a lot of good memory. So over the last 5 years, and most of the time we traveled around. The one time Michigan, Ann Arbor, another time Beijing, and about the delegation size every year they got to increase from the very beginning 70 and to more than 100. So everyone enjoyed, also learned a lot from a collaboration between the Michigan and the PKU. So okay, back to the focus of this meeting. I'm going to introduce something they have been done specifically in my lab, in my research team. So as I put here, so I will present you roughly like a genomic analysis and a molecular dissection. So we do a lot of like genomic instability, cell cycle regulation, and do a lot of molecular signaling work. But since I came back from Pittsburgh, back to Beijing, and then we picked up the cancer type called like esophageal squamous cell carcinoma, which typically we call it with China feature because for example, like US doesn't have much esophageal cancer, but even some people have is like adeno, and we have squamous carcinoma. Next slide. Okay, actually back to the 80 years ago, and we made the first report to identify genomic alterations in ESCC. We first time reported like 8 significant mutated genes, including the famous one like P3, CDK, N2A, RB, the PI3K, Notch. So only one, actually we made the first report is the gene with oncogenic property is FM135B. So right now, and we develop a lot of resources to study this one. So during these studies, and in addition to the significant mutated genes, and also we find a lot of the somatic aberrations they connected to the Wind cell cycle, Notch pathways. Another is associated with lifestyle. So we during this work we only confirm is very closely associated with alcohol drinking, but we didn't pick up like with smoking. So later on, and we found is like statistically maybe also related to the smoking too. Next one. Next slide. Okay. In addition to that one, also we did another things is we pick up like a multi-region sequence, and then do the special to check the spatial heterogeneity. And in this one, we pick up the different samples to do the genomic analysis from primary tumors, also metastatic tumors, pick up the lymph nodes, and even primary tumors, we pick up multiple sites in one tumor, and to see how is the genomic alterations during in the different species. And use this one, and we found several, we call these action actionable targets for subset of ESCC. Next one. Next slide. And most recently, and we enlarged our sample size. This is like 500. No, not yet. Back to that one. Back forward. Slide, please. Yes. Okay. These 5. Okay, maybe is that. Okay. Using the we call it the largest like sample size, and we did further analysis for the genomic.
Next slide, please.
Not yet. Okay. They actually is a previous one, because I think it may be a slide they set up like it's automatic. This one. Okay, this one, and then yeah. No, next one. No. Make forward. Next one. Next one, please. Okay. Use this one, and we divide. Okay, divided like 3 different types of like a biomarker, which is associated with clinical features, and can make can do some they can predict like therapeutic the efficacy for the immunotherapy. Okay, in addition to the whole genome analysis, this is what we did. This one we tried to figure out what is a crosstalk between tumors and the tumor microenvironment. And then we focus on the DGK alpha and FAK axis, and another one is NOX5. And we found that DGK alpha can activate FAK the pathway, and also NOX5 also is got involved too. They function as an oncoprotein to oxidize and activate the one important like a kinase called SOC kinase, and then to make tumor cells more malignant. And in addition to that one, and this pathway also regulate the release of cytokine. This cytokine can release from tumor cells and interact with macrophage and with the fibroblast, and to collaborate with this macrophage and the CAF, and make this environment more suitable for the tumor cells. So next slide, please. This is what we did, and then we think about next slide, please. So we think about in the future. Also, we like to see that try to identify the slides is a little bit messed up. Try to identify the new functions for that the signaling regulation, the pathways. Also try to figure out the more molecular mechanism and what is interaction between the tumor microenvironment and also the interaction between the tumor cells. So also this one definitely, because the tumor environment and then they play important roles for the cancer immunotherapy. Okay, this is another work. We found the MAGC3, which is amplified, gene is amplified, is membrane protein. One way it can activate STAT3 and regulate the EMT. Another interesting finding we got is because the IF interferon gamma can activate this one, then activate the STAT3, STAT1 pathway, so regulate the PD-L1. So we think this the pathway also is going to be like a target, or we activate like a tumor therapy. When we block this one, and they increase the immunotherapy sensitivity. Next one, please. Next slide, please. Then this is what we believe, according to what we found, and this MAGC3 probably can provide avenue for the modulation of interferon gamma-related. The inflammatory disease also could be the prognostic marker, and also can provide this biomarker for the therapeutic target. Next one. So quickly, this is another issue. What we did is the cancer caucasia. We know that a lot of people die during this caucasia because caucasia is pretty complicated. But when people suffer to this stage, then a lot of people will going to die. But unfortunately, there's no specific drug to treat caucasia. Next one. So what do we found? Next slide, please. Next slide. Okay. Then what we did, we first deliver the ESCC-induced caucasia, the mouse model. Then we identified, we isolated. We think the tumor cells will release some important element. They regulate, for example, regulate the muscle cells to eliminate the muscle cells through the cell apoptosis. So we identified the extracellular vesicle derived from tumor cells which contain P4HB, and these P4HBs, the one gene that can regulate PHGDH and BCL2 pathway. Then the end point is they can activate the caspase-3 pathway, and then so right now we use a P4HB inhibitor. They can greatly inhibit apoptosis. Then we raised a few questions. Next one. This is next one is going to be last one. Next slide. Okay. Key point and in addition. No, not yet. Please back one slide. Okay, this is what we think about. In addition to these P4HB affect caucasia, regulate the muscle, and make muscle disappear through the apoptosis. And then another thing is how to regulate the metabolism of the fat. Like lipo metabolism. This is something we are doing now. Another issue. Next slide. Next slide. I point out. Next slide, please. Okay. They help. Okay, just forget about that one. Then right now the important issue we are studying now is how caucasia to affect immune system, because when this caucasia induce that exhausting situation for the patient, definitely they will destroy the immune system. So how these caucasia tumor cells release something like a P4HB or some like factors that regulate immune system. This is an important issue, and we think right now we are working on this one. So I will stop here. And then they wait for the discussion with you guys. Thank you.
Thank you, Dr. Zhang. And we have a question from our audience, from Jian Guo. And
we're going to go through all of the talks first and then have questions and discussion. Otherwise, I think we'll be running short of time. So
okay.
I think we'll have questions for everyone at the end.
All right, let's do that. Okay, I would like to introduce the next speaker then. Next speaker is Professor Huang. Professor Huang is the chair of Peking University Institute of Hematology, director and professor in the Department of Hematology. And he also holds many titles, including the director of the National Clinical Research Center for Hematology Diseases in China, and also the chairman of the Chinese Association of Hematologists, and the chairman of the Committee of the Experimental Hematology, etc. And Professor Huang's research focuses on haploidentical hematopoietic stem cell transplantation, the prevention and the treatment of transplantation complications, the stratification and the targeted therapy of leukemia. And Dr. Huang has published more than 300 peer-reviewed papers. And let's welcome Dr. Huang, please.
Dear Professor Zhang, thanks for your nice introduction. So I am a hematologist and also a clinician. So I focus on the treatment of leukemia and also some translational research. Next slide, please. Okay. Because hematopoietic stem cell transplantation is one of the most important therapy for leukemia. But before 2000, there's a short of donor. So based on GCSF and ATG, next slide, we established a series technology. Next slide, please. Forms are hypo system, hypo transplanted system. Please, next slide. And because this system, so hyper-identical transplantation has become routine, so everyone have donor for transplantation. Next slide. Please, next slide. So because everyone have donor, so the number of transplantation in our institute increased continuously. Last year, we finished more than 1,200 cases of allogeneic transplantation. I think this is the largest transplant center. So that means since to want to promote our cooperation, I think this is a huge number of patients. So this is a big platform for cooperation. Next slide, please. Okay. However, relapse remain the first least cause after transplantation. Once the patient relapsed, then the result was very poor. So I think the key point is that we need to optimize the graft versus leukemia effect after transplantation. Next slide. To do this, we actually developed several strategies. For example, we modified traditional donor lymphocyte infusion to increase the anti-leukemia effect via decrease the graft versus effect. So modify the donor lymphocyte infusion can reduce the relapse after transplantation with relatively safety. Next slide, please. And also, okay, next slide. Also, we also use interferon alpha for treatment of MRD positive patient to enhance, to promote the graft versus leukemia effect to decrease the relapse rate after transplantation. Please, next slide. And also we use CAR-T for MRD positive for MRD patient after transplantation and decrease the relapse rate, increase disease-free survival. Next, please. And also we develop a total therapy strategy, including MRD monitor and modified donor lymphocyte infusion to treat high-risk relapsed leukemia and achieved 50% long-term disease-free survival. Next slide. And our data also confirm that actually hypoidentical transplantation have significantly stronger graft versus leukemia effect compared to matched simple donor transplantation. As demonstrated here, MRD after hypoidentical transplantation can decrease significantly faster compared to matched simple donor transplantation. This translates into better lower disease-free survival and better lower relapse rate after hypoidentical transplantation. And the animal model confirm hypo have a stronger graft versus leukemia effect. Next slide. And also, I know the college in Chicago also use interferon alpha. Use PEG interferon alpha for treatment or relapse after transplantation. I think it might be we can some kind of cooperation in this field, especially for the understanding the mechanism of interferon alpha after transplantation. Next, please, next slide. And also, I know the college in Chicago also use the RNA sequence of technology to study the regulation of T-cell immunity. I think we also can share this kind of a platform to do some kind of cooperation. Next slide. Next, I think this will be the last slide. Although because our college of in Michigan also for interested in leukemia, but actually in terms of joint program, I think we can have some protocol, join the protocol for in the field of graft versus leukemia effect. Next. Okay, thank you for your attention. I think after I stop here, maybe after all the talk give their presentation, we can some kind of cooperate discussion.
Right, thank you, Dr. Huang. Our next speaker will be Professor Fuping Yu. Professor Yu obtained his PhD degree in immunology at Peking University. Then he received his postdoctoral training in Yale University School of Medicine. Now he works in our Department of Immunology, and he's a researcher in the 100th Talent Program of Peking University School of Basic Medicine. And his research focused on the immune regulation mechanism during the occurrence of viral infection or in colorectal cancer and gastric cancer. And his work has been published in Nature Immunology and the Cell and the Science Signaling, etc. And let's welcome Dr. Yu, please.
Okay, thank you very much for introduction. It's my great honor and pleasure to introduce the recent advance of our work. My lab basically work on investigating the aberrant pattern recognition during viral infection and tumorigenesis. Next, please. It's believed that the immune system is crucial for eliminating the invading pathogens and surveillancing the inner abnormal such as tumor. The immune system include innate immunity and adaptive immunity. innate immunity serves as the first line of host defense, can recognize the invading pathogens very quickly, and by employing a cluster of pattern recognition receptors, PRR, as well as recognizing the inner abnormal, such as tumor cell. The PRR, in addition to recognize the pathogen-associated molecular pattern, PRR also can recognize the damage-associated molecular pattern, DAMP. Next, please. So my lab are focusing on clarify the mechanism of innate immune signaling transduction during viral infection and tumorigenesis. Recently, we identify a nuclear viral RNA pattern recognition receptor, SFA. SFA can sense the viral RNA in the nucleus and regulate the chromatin remodeling and initiate the transcription of antiviral genes. Recently, we also found not only the viral RNA, but also this new synthesized host RNA also can trigger the oligomerization of SFA and mediate the chromatin remodeling. Currently, we are working on how the chromatin remodeling directs the cell identity and the cell death during viral infection and tumorigenesis. Next, please. Firstly, I want to introduce briefly about the work about the COVID-19. It's well known that the asymptomatic carrier and the cytokine storm in the COVID-19 is two significant features of COVID-19, which caused the pandemic of COVID-19. As for the asymptomatic carrier, they can transduce they can mediate the transmission of virus in the population without showing any symptom. That means at the very beginning of infection, in these people, the innate immune pathway was suppressed. On the other hand, in the severe COVID-19 patients, the cytokine storm was initiated by the overactivation of innate immune system. Next, please. So by employing the animal model, including the mice model and the research macaque model, the monkey model, we identify the innate immune signature during the SARS-CoV-2 infection. We identify the pump pathogen, also the pump and the DAMP initiate abnormal innate immune signaling during SARS-CoV-2 infection. Firstly, it's the S100, A8, and A9. They form the heterodimer function as alarming, can directly interact and initiate the toll-like 4 signaling. We observe that there are a thousand-fold induction of this alarming in the lung of rhesus macaque and the mice. Additionally, we found the spike protein of SARS-CoV-2 can directly interact with TOR4 and mediate this signaling transduction. Actually, the spike protein initiates this abnormal immune response. The S100 protein functions as an accelerator or amplifier to mediate, to accelerate or expand this abnormal immune response, because it's well known that tolerance 4 actually as a PRR can recognize the gram-negative bacteria, but here we found the iron virus SARS-CoV-2 initiate this aberrant immune response and also promote the expansion of a cluster of a number of immature neutrophil, and this kind of immature neutrophil greatly induce inhibit the function of leukocyte, and this kind of neutrophil probably function exert the similar function of MDSC during during the tumor genesis. Currently, we are working on to figure out if this kind of immature neutrophil is a kind of can exert a similar function of MDSC, and under this abnormal immune response also can explain why the most of the severe COVID-19 patients exhibit the sepsis symptom. Next, please. Secondly, I will briefly introduce the work about the phagocytosis and anti-tumor immunity. It's well known that the phagocytosis is critical for enhance the anti-tumor immunity. We here found a deopicuidinase OQD1 can enhance the iron transplantation and mediate the phagocytosis and the release of ATP. And this ATP can activate the CD4 T cell. But how the ATP, CD8 T cell, and mediate the tumor killing. But how the ATP activates CD8 T cell is unknown. We are working on this now. And secondly, during we work on the syncytial formation during coronavirus infection, because you know in the clinical data, the syncytial formation was observed in the lung of COVID-19 patients. So we found that this syncytial formation is mediated by phagocytosis. But we found this kind of phagocytosis only occur in the macrophage or the tumor cell related to macrophages. So nextly, we try to figure out if other kinds of tumor cell can undergo the phagocytosis after coronavirus infection. So we perform screening by using melanoma, and we treat the cell with erasing phagocytosis agonists, and we construct a CRISPR library containing all the membrane protein around 5,000 protein and 5,000 genes and 10 individual guiding RNA for each genes. After the screening, we observed some gene can inhibit phagocytosis and some gene knockout can promote apoptosis. Here we found of the RTB4 gene knockout, the coronavirus MHV can induce massive cell death of melanoma. And consistent with the clinical data, the RTB4 in the cancer patients, the RTB4 high expression RTB4 can cause poor prognosis. Next, please. Lastly, I want to mention about the story about inflammation, tumor, and aging. The colitis associated cancer is promoted. Promoted by the inflammation, and the bad inflammation can also induce DNA damage. Here we found that this bad inflammation downregulate ER4 through a epigenetic mechanism. Interestingly, we found the montamorelonite powder can rescue the downregulation of ER4 and in turn rescue the DNA damage repair machinery. Here you can see the patient regularly use this powder can greatly inhibit the progression from UC to CAC. Lastly, I want to briefly introduce the story about the inflammation and aging. Inflammation and aging. It's been very well characterized that the LAMI-A mutation can cause the HGBS, a typical progeria. But here we found in 3 Chinese family, a homologous mutation of LAMI-A R527C can cause inflammation-driven progeria, and he is one of the family. Different from the typical progeria, all patients exhibit a much severe autoimmune symptom, and mechanistically we found the gene damage, the damaged DNA function as a dump can activate the DNA sensing pathway and promote the inflammation. After we knock out M2, a DNA sensor, it can completely rescue the early death of the mice. And the C176, a small molecule, actually is an inhibitor of cGAS-STING pathway, can rescue the cell synthesis of the mutant cell. Currently, we are working on evaluating the safety and the delivery strategy of this drug in the animal model. Okay, that's it. Thank you very much. Thanks.
Thank you, Dr. Yu. And next, I will give this moderator position to Dr. Max Wicha, who is an old friend of us, and also Dr. Wicha, one of my good friend, Liu Sulin, was actually from your lab, and she's doing great in China. So Dr. Wicha, please.
Yes, and I hope to be able to visit Sulin soon. So our first speaker from the University of Michigan Cancer Center is Dr. Weiping Zhou, who is really a leader in the field of tumor immunology. He's the Charles B. de Mancley Professor of Pathology, Immunology, Biology, and Surgery, and he serves as the co-director of the Hematopoiesis and Immunology program in the Cancer Center. As I said, he's really made a number of seminal contributions in the field of tumor immunology and has published almost 200 articles, of which over 40 are in Nature, Science, and Cell journals. So, Weiping.
Right. Thanks, Max, for your introduction. I hope you can hear me well. In fact, as this is a bridge conference, I prepared a few slides. It's kind of introduction, what we are doing here. So you can see from my slide, I will really focus on some novel mechanisms in the lab, epigenetic mechanisms and metabolic mechanisms. Next slide, please. So the purpose, what we are doing, not only to understand the basic mechanisms of cancer, including immune responses, but more importantly, we want to see translation, means how we are going to treat the patient. So if we review our human history in cancer therapy, we have several milestones. So number one, surgery, then radiation, then hormone therapy, and we have chemotherapy and targeted therapy. And finally, at these days, we have immunotherapy. So the panel has already asked, what's next? So next slide, please. So when we see those milestones in our human history, each milestone is really behind of the basic scientific discovery. So what's next in terms of the therapy? It's really dependent on our basic understanding of immune responses and the tumor microenvironment. So my group has been working on cancer microenvironment for many, many years. We have several research directions here. One is immune suppressive networks. The example is the PD-1, PD-L1 blockade. And then we worked on cancer epigenetic, and the cancer epigenetic mechanism related to immune modulation in the tumor microenvironment. We also worked on the immunogenic pathways and the autophagy pathways, and the way how the immunogenic pathway and autophagy pathway regulate immune responses and immunotherapy sensitivity. And then in the recent years, we have spent quite a lot of time working on metabolism and how metabolism affect tumor immunity and therapy. So in terms of the mechanism, metabolism, we have high interest in multiple pathways, including glycolysis, oxidative phosphorylation, amino acids, lipids. We have all interest in those areas. Move on, please. Next. The major focus in the recent years in our group is on metabolism. The thing is, the cells, including immune cells, tumor cells, fibroblasts, any cells in the tumor microenvironment are exposed to the microenvironment, therefore are exposed to the nutrients and the metabolites in the microenvironment. So those components will affect the cell status, including their phenotype and functionality, and obviously the response to different types of therapy, not only immunotherapy. Next. So the major focus in this case, because metabolism is a huge area, I would say we have 3 areas. So one is ACLC family members. These particular family members, we have about 500 members, let's say. It's very poorly defined, either in metabolism or in immune responses in general. We have high interest in that. The second is fatty acids and lipids. We have high interest. And the third one is the crosstalk between epigenetic mechanisms and metabolic mechanisms, and particularly the relationship with tumor metastasis and the therapeutic response. Next. So that's all. I guess we can have some discussions in that space. And generally, as Max introduced me, in the cancer center, we have this cancer hematopoiesis and immunology program. I'm the program director along with Baban Reddy. We'll see the interest from our friends in Beijing. And if we have interest in different areas, not only just related to my own work, but also the work in at the University of Michigan in other programs, I surely will bring those to our friends here. Thank you.
Thank you, Wei-Peng. Our next speaker is Dr. James Moon, who is the J.G. Searle Professor of Pharmaceutical Sciences and a Professor of Biomedical Engineering. Dr. Moon is interested in the development of therapeutics and diagnostics at the interface of immunology and engineering. And as you'll hear today, he's developing new drug delivery systems that can enhance targeting of antigen and adjuvants to lymph node organs, thereby multiplying the effects of the immune therapies. It will tell us also about the nanoparticle vaccines that he's developing as a strategy. So, Dr. Moon.
All right, thank you, Max, for a very kind introduction and invitation to share our work here. So my lab is based on biomedical engineering and pharmaceutics. We are developing new therapeutics to deliver immune activating agents for improving cancer immunotherapy. So first, the project that I'd like to discuss is on a therapeutic vaccination. Next slide, please. As you probably know, there has been a very exciting work performed in various parts of the world that have shown that when you take tumors out from patients and do DNA, RNA sequencing, you define the multiple mutations. And because patients have accumulated mutations over decades, in most cases, patients would have specific mutations that are specific to each patient. And what that leads us to is identification of a novel antigen called the neoantigens that are displayed on cell membrane. So there have been a lot of efforts in various parts of the world, including China, to develop personalized cancer immunotherapy once you use these neoantigens to target everyone's cancer mutinome. In my own lab, we utilize nanoparticle systems to deliver these neoantigens for generating very strong cytotoxic T-cell responses. One platform we are developing is a synthetic high-density lipoprotein nanodisk that's 10 nanometers in size. We engineer the system so that after subcutaneous injection, they enter lymphatic vessels very efficiently, delivering these neoantigens and adjuvant molecules to lymph nodes. And that leads to uptake by dendritic cells, induction of very potent T-cell responses. Next slide, please. So in this example, we used a single neoantigen from colon carcinoma model MC38. And after vaccination in mice, we get up to about 30-fold induction in the CD8 T-cell responses compared with the other leading adjuvants in clinical trials. So next slide, please. When we use this therapeutic vaccination in combination with immune checkpoint blocker, anti-PD-L1, we see up to 80 to 90% tumor eradication rate when we use our nanodisk to deliver single neoantigen peptide plus immune checkpoint blockers. And that's much better than injecting free naked peptide as a neoantigen vaccine in this model shown in red. Next slide, please. We have done this work in multiple murine tumor models. This is a work done in collaboration with the Maria Castro in the Cancer Center here in Ann Arbor, where we used glioblastoma multiform neoantigens. So when we have a multiple neoantigens, we put them on separate nanodisk and pull them right before injection in a cocktail approach. And in an orthotopic glioma model, when we combine 3 different neoantigens plus immune checkpoint blocker anti-PD-L1, we saw very robust induction of neoantigen specific CD8 T cells in PBMCs. Next slide, please. And then the T cells are infiltrating into the brain, as shown by this flow cytometry data set. Next slide, please. And this results in tumor eradication in up to about 35% of cases shown in blue. And if you use a conventional free mixture of a peptide and neoantigens with immune checkpoint, you don't have any survivors, as shown in red. Next slide, please. And we can also do a tumor cell rechallenge on the contralateral hemisphere and show 100% protection, showing that this is a fantastic personalized approach for eliminating primary tumor, as well as establishing long-term immunity against the tumor relapse. Next slide, please. And we are doing some work with the non-human primates to identify the lead candidate for clinical translation. In particular here, we were screening different adjuvant molecules to combine with our nanodisk vaccination approach. We looked at TLR3 agonist, poly-IC, as well as TLR9 agonist, CPG, with the 2 different formulations, CPG-B and C. And after 4 monthly injections in rhesus macaques using an HIV gag antigen, we see very robust induction of CD8 T cells in lung lavage fluid with a Hilton poly-ICLC combination. Next slide, please. And we can also prime these monkeys with the nanodisk platform and then boost with the advector. This advector is what's being used in Janssen's COVID-19 vaccine, but it's engineered to express the HIV gag antigen. So what you can see in blue is that monkeys that are primed with the nanodisk Hiltonol and then boosted with the advector mounts very strong CD8 T cell responses. So we are moving forward to clinical translation with neoantigens for personalized cancer immunotherapy. Next slide, please. And this is another project where we are trying to engineer new ways to deliver STING agonist for cancer immunotherapy. Next slide, please. This work was inspired by actually work coming out from Peking University from Dr. Zhang Fan's group, who reported that manganese can interact with the STING receptor. What we found in our work was when we combine cyclic dinucleotides, the STING agonist with the manganese, we see very potent synergy. And when we couple that together into nanoparticle formulation, we can now do intratumoral as well as systemic injections for co-delivering both agents to dendritic cells and macrophages, leading to very strong immune responses. Next slide, please. So we engineered these basically liposomes delivering both cyclic dinucleotides and manganese. And these nanoparticles are about 170 nanometers. And they very much promote cellular uptake, shown in red, much better than injecting free naked STING agonist. Next slide, please. This results in very potent STING activation and type 1 interferon secretion from dendritic cells, shown in red. Next slide, please. And after IV infusion in tumor-bearing mice, what we see is that these nanoparticles that we call CMP are avidly taken up by dendritic cells, macrophages, MDSCs within the tumor microenvironment. And we see much better delivery compared with injecting free naked STING agonist, the CDA, shown in blue. Next slide, please. And this results in activation within tumor microenvironment, leading to increase in M1 macrophages. Next slide, please. Dendritic cells and activation of CD8 T cells and NK cells. Next slide, please. And we also see a decrease in MDSCs showing very potent immune activation. Next slide, please. We have shown the efficacy in multiple murine tumor models, but this is a key example in the MMTB-PYMT. This is a triple negative breast cancer model that's very hard to cure or treat using conventional chemo or immunotherapies. Next slide, please. So here are these mice developed tumors starting 40 days of age in multiple mammary fat pads. Here we waited until very late stage on day 75 and gave it 6 rounds of the IV treatments with our STING activating nanoparticles. And those mice are significantly inhibiting tumor growth compared with pre-naked STING agonist that's not showing much tumor inhibition, shown in the top. So we are very excited about these approaches where we can deliver STING agonist in a synergistic manner with the manganese for activating immune responses. Next slide, please. We are very interested in a collaboration. Hopefully we can start a new collaboration with the labs in Peking University on a therapeutic cancer vaccination, STING activation, as well as other ways to deliver immune stimulating agents for cancer immunotherapy. Thank you for your attention.
Thank you very much. And our final speaker will be Adam Courtney, who's an assistant professor of pharmacology. His research focuses on how T cells make decisions that affect their behavior. He takes an interdisciplinary approach that applies immunology, chemical biology, and genetics to investigate T cell signaling pathways. His research interests include signal transduction in the immune system, T cell signaling pathways, and immunotherapy strategies. So Adam.
Yeah, thank you. This is my first time participating in the Joint Institute, so this is very exciting for me. And I'm looking forward to telling you a little bit about how we think about things in my lab and some of our experimental approaches. And so we mostly think about things from the T cell perspective and how T cells signal to make decisions. So next slide, please. Yeah, and so if we think about this in the context of cancer, where cancer establishes this vicious cycle of immune evasion, so T cells can't do their job, what we're kind of thinking about is how we can restore the signals T cells need to mount an effective anti-tumor response. And so there's three main signals that we think about, and cancer dysregulates these. One is the quality and presentation of antigen. The other is co-stimulatory pathways and co-inhibitory pathways that become dysregulated. And the third is the instructive signals provided by cytokines that can become depleted in a tumor or are replaced with cytokines that maybe misdirect the immune response. So thinking about how we might restore those signals. And so the goal of that would be to rescue a cellular response at the T cell level, but then also maybe more further long-term, more phenotypic response in T cell populations. And so, I think we've heard of a bunch of different strategies people use, and the two main ones that we think about are, or compare and contrast, are receptor-targeted strategies like those that might target inhibitory co-receptors, or basically, or agonists that target co-stimulatory receptors, or James Moon talked about vaccines that maybe can be recognized by a T cell antigen receptor. We're also thinking a lot about intracellular targets. Both approaches probably have their pros and cons. So just thinking about how maybe some of these pathways might have common intracellular mediators, or might escape redundancy if you have multiple inhibitory receptors. Maybe targeting a common negative regulator inside the cell might be a good strategy. So that's sort of how we're thinking about things, and some of our science is sort of evaluating these ideas. Next slide, please. So this is just showing the three signals that I just mentioned and that we're thinking about quite a bit. Signal one is provided by the T cell antigen receptor. I won't go into a lot of detail about this, but basically it requires tyrosine kinases that phosphorylate proteins that associate with the T cell antigen receptor to basically propagate signals. And this is necessary for T cell antigen recognition and T cell response. Signal two is provided by co-stimulatory receptors. And then signal three is another pathway that we're very interested in, which is mediated by cytokines and JAK-STAT signaling pathways. And so thinking about some of these intracellular mediators, such as STAT proteins, LCK is a tyrosine kinase, and how we might target these to sensitize T cells or provide signals inside the cell as an alternative strategy. And our goal here would be to rescue impaired signaling that can happen within the tumor and restore this cellular response, and basically perhaps rescue a functional phenotype that would allow a T cell to resume its attack on cancer. Next slide, please. So we take a very bottom-up approach in my lab, and so I just wanted to highlight some of the challenges we face and maybe see what people think about that, and highlight some of the systems we're developing. So when we think about signaling, a lot of times we're talking about protein regulation, and so a major challenge here is identifying a target to pursue, and then how are we going to manipulate the activity of that target, either using an antagonist or an agonist or genetic modification. And then we need to sort of evaluate how we might, is that target doing something to the signaling pathway in a context that we think might be relevant. So we have to come and challenge the T cell with different signaling inputs, and then that might be relevant, and then somehow have an output that informs what we might want to do in a cell-based assay, looking at some sort of function within that the T cell can perform. And so we think a lot about ex vivo systems, and especially in the setting of cancer, recapitulating hallmarks of the tumor microenvironment in ex vivo settings, so we can sort of look at how these perturbations are affecting T cell function in a relevant system. And probably the biggest leap we have to make is then translating that to an in vivo setting, and thinking about, if we are manipulating a pathway and we just go directly in vivo and we sort of try and do this in vivo, how do we know if we're affecting that pathway in a physiologic setting? And so thinking about that a little bit. And so this is sort of how our pipeline is in the lab and how we are approaching things. And so trying to integrate these different systems is, I think, a challenge that we're starting to appreciate, and something that we're working on. And so maybe I'll walk you through a short story. So can we go to the next slide, please? So one thing we became interested in, this is work that I performed while I was at UCSF, was we found that if we inhibited CSK, so this is a negative regulator of the tyrosine kinase LCK that's involved in TCR signaling, we found that we could sensitize T cells to low affinity antigens. So shown in those two plots are different doses of a peptide antigen, and looking at the proportion of activated T cells as assessed by upregulation of an activation marker CD69. And what you can see is that If we come in and modulate the activity of this kinase using an inhibitor of LCK or inhibiting an inhibitor of LCK, so indirectly activating LCK using the CSK inhibitor, what you can see is that the response of these T cells to a high affinity antigen, in this case ovalbumin peptide, is not really changed much. But when we look at this low affinity antigen, a variant peptide, we can really sort of titrate the ability of the T cells to respond to these low affinity antigens by modulating the TCR signaling pathway. And so if you actually hit it, I think there's a pop-up here, I forgot about that. The way we think about this is that we're sort of shifting the threshold for T cell activation where, in the case of a high affinity antigen, these inputs are not necessarily changing anything, but for a low affinity antigen, we're making the T cell more reactive so that it can respond to it. And we think that this might be beneficial in the setting of cancer if, a tumor antigen becomes downregulated, or it's not as great as, for example, an antigen that might be presented by a completely foreign organism like a bacteria or a virus. Next slide, please. So to try and do this in a setting that might be relevant to cancer, we're sort of trying to develop an integrated approach where we've been developing these assays where we can do this high content imaging using reporters of TCR signaling. So in this case, using a NIR77 GFP reporter that responds to the TCR signaling pathway. So what you can see, it might be a little bit hard to see, but we can do this in sort of a high throughput format where we set up co-cultures of T cells and cancer cells. In this case, MC38 cells that express a model tumor antigen, ovalbumin. And we can look at these T cells light up and turn green when they recognize this tumor antigen in the MC38 cell co-culture. So we'd like to be able to sort of establish this as a platform, maybe incorporate suppressive milieu to see how that might affect antigen recognition and what our perturbations to signaling pathways might do to overcome those suppressive features. And then the idea here is that this reporter can be used in vivo. And so shown in the right three panels are just T cells, or I guess hematopoietic cells that were isolated from an MC38 tumor, gated on CD45, and we look at CD8 T cells. In the histogram, what you can see, this is just a polyclonal T cell response going on within the MC38 tumor, and what you can see is the NIR77 GFP reporter. There's a great deal of variability in the level of GFP expression amongst T cells within the tumor, sort of reflecting perhaps their ability to sense antigen within the tumor microenvironment. And so what we'd like to do is sort of build on some of these platforms to try and basically look at how some of the therapeutic strategies we're thinking about that modulate signaling pathways, for example, JAK-STAT signaling or tyrosine kinase signaling might play out in both the 96-well plate all the way to the tumor. So next slide, please. So we're very interested in identifying new therapeutic strategies and, looking at, modulators of these targets. And then, so what we're really trying to do here is establish some ex vivo assays to test these treatment conditions that can be translated all the way to a mouse tumor model. And, I think, so we think a lot about tumor models and also how we can sort of incorporate some of the stuff we're doing into T cell based therapies like CAR T cells. So that's basically what I wanted to share with you guys today.
Excellent. I think, again, we've heard exciting proposals, descriptions of research from both institutions, and we're going to now open this up for a panel discussion. So please send your questions into the chat room, and Ning and I will look these over and then sort of submit the questions. We'll ask them. I think probably would be a good idea on the screen to just raise your hand, and then we can call on you for this. Maybe I can start actually with a question from Dr. Zhou to Dr. Zhang about esophageal cancer. He asks, how can we use genetic tools to define the immune nature of esophagus cancer and immunotherapy sensitivity? Are there any obvious immunological, etiological, or therapeutic differences between esophagus cancer in China and the United States?
Okay, regarding this question, I think that gives some points. First of all, there is no any effective therapeutic approaches to the esophageal cancer. I mean the squamous carcinoma. So the no targeting therapy right now. They also use the platinum the type of the therapeutic agent. So right now, two things we are used. One is definitely use the PD-1, the PD-L1 to test the whether, because whether it can work on the esophageal cancer. So with the collaboration with the people, and we did did like a T cell microenvironment, the T cell the sequence, and to see the whether they have a healthy environment to using these approaches. So this is currently what we did to determine the whether the microenvironment, whether they have the pretty good the level for the T cell to respond to the PD-1 the PD-L1. This is one thing what we did, and also we are doing now to enlarge the samples. The second issue that is more specific, just I mentioned it, like MAG is 3, which it's we found is amplified in the esophageal cancer, and this one because they in the presence of interferon gamma, they also can activate STAT1, and then they regulate the PD-L1 expression, and then using the inhibitor for this MG3 and plus the antibody, the PD-1 antibody, and we generate a pretty good response. So we think this is one of the target probably can use for the clinically they use the immunotherapy for the esophageal cancer. Back to you, Max.
Okay. I think a general question that probably for any of our speakers is, what are the current limitations of immune checkpoint blockade, and what do you think are the major opportunities in the future to go beyond that? And then in particular, are there areas of commonality that you've seen here between the University of Michigan and Peking University that we may be able to exploit? Anyone want to take a stab at that?
Yeah, I can say a few words. Can you hear me?
Yes. Mm-hmm.
Yeah, I can say a few words about the first part because there are several questions behind actually. So I think I know better about the checkpoint part, so I can say a few words about that part. So in general, right now, all anyone is talking about in the medical field, if you say cancer therapy is immune checkpoint. So basically, so far we talk about PD-1, PD-L1 blockade, and NASDAQ and CTLA4 basically. So there are many questions, of course. The majority of the patients are not responsive. A small fraction of the patients are responsive. So that's everybody knows basically. And we recently even know, not published data from my own group, we know a small fraction of patients that when they receive the immunotherapy, they get worse. We are working on that, why those patients are getting worse. So this raises the issue. It means immunotherapy does not work in everybody, and sometimes it's even generate a detrimental effect. So the question comes back to the point that we need to understand much better the tumor microenvironment, and also not only each type of cancer, it's not a one disease, multiple diseases, but also we need to understand each individual patient in many ways. So in this case, the next approach is, that's why I asked Jimmy the question, how can we use different technology tools, including genetic tools, to understand better the nature of the immune responses in the tumor environment? Can we use those technology, and you use those technologies to get the patient therapy? So that's probably the way to go. So understand the immune responses interaction with different cells. We don't have a fix here, you know. So that's one way to go. Another way to go also, we have to understand that and identify different therapeutic targets in the space of immunotherapy as well. So as I said, now you only talk about PD-1, PD-1, and CTLA-4. There are some other mechanisms, maybe important target we don't know. So people are working on this. Maybe new combinations and a new target will be useful in different types of disease. That's the hope.
Yeah. Can I add some points on that one?
Oh, go ahead, please go ahead.
Okay. About 2 weeks ago, I went to the another city, Qingdao, for the cancer conference. The people use because of the terminal patients. Terminal patients, there's no any the usable approaches for those patients. And then after like an ethical issue discussion, so the people use just to take a try. They use for terminal patient, they use the PD-1, the antibody, and the plus use the targeting therapy against the angiogenesis. They really they did find some effective the therapeutic the response. Then they go back to check do the RNA-seq, back to like Weiping the question. Then pick up a couple of key points. Because like a regulator, we call it the master regulator. We never define this as a biomarker for selecting, like precisely selecting the patient for the treatment. This is just take a try. But they try to like about 10 people, they pick up 3 people is a very, very responsive to the immune response. But you check the PD-1, PD-L1 is not high. The level is not high. And if you, according to the current outline, you may not pick up this patient, but because this is a terminal patient, they want to use this one. And then they tried this, and then they found that they did find some like a new target. Right now, use this new target, they enlarge the sample, the patient to see do the could be the indicator to pick up the effective patients for the for this kind of a treatment. Yeah, some information I add on.
We got a few questions about tumor heterogeneity, and I'd like to kind of ask that in a little bit a different way. So a couple of the speakers addressed the issue of genetic targeting and genetic heterogeneity in designing therapies, particularly Dr. Moon talked about looking at tumor heterogeneity. I wonder if anyone would address the issue of epigenetic heterogeneity, and particularly one area that we've been interested in our own lab, and that is stem cells within cancer. And how well is immunotherapy currently targeting stem-like cells in tumors? And does this account for some of the resistance and relapse, and how could we do better in doing that? Anybody want to tackle that? No. James, you want to, I know you work on that. You want to talk a little bit of the Dr. Moon, a little bit about targeting stem cells and tumors with immunotherapy?
Yeah. So in collaboration with Dr. Max Wicha, we've been finding transcription factors that are found in cancer stem cells and using that to engineer therapeutic cancer vaccines. So for example, we can target NANOG or SOX2 expressing cells using our nanodisk delivering those peptides and generate CD8 T cells to go after cancer stem cells. So in mouse model of a melanoma, it's been working well, and we've been trying to show their efficacy in other tumor models. So I think it'll address the question whether cancer immunotherapy can be broadened by targeting the root source of the cancer cells, the cancer stem cells. So we have a lot of work to do, but I think we are getting there. Yeah.
Maybe you or others could also then talk about, a few speakers talked about the STING pathway and innate immunity. How can we best coordinate or add innate immune therapies to adaptive immune therapies, and is that going to be something important in the future? Perhaps, Dr. Yu, you talked about the STING pathway and things that you're looking at there. How do you see combining STING therapy with checkpoint blockers and things like that?
Okay. It's well known the STING pathway can mediate the robust induction of type I interferon, and the type I interferon is can promote the activation of dendritic cell and the T cell. And probably I think currently the most important challenge I think is the side effect. The uncontrolled the type I interferon is harmful for our host. For the delivered agonist of STING or collaborate with the or combine with the mechanisms just James Moore showed. I think the effect the efficacy is very good, but the side effect I think currently maybe a big obstacle for combine the STING innate signaling to the checkpoint treatment. That's my point.
Good. And Dr. Huang, it was very good to hear about the leukemia service that you have and the large number of transplants. As you know, University of Michigan also has a very major program in bone marrow transplant and CAR T therapies. I wonder if you could talk about some of the things that you're doing in graft versus host disease and some of the areas then that we might be able to do together, looking at both graft versus leukemia versus graft versus host disease, looking at distinctions in that. You see areas there for collaboration?
Yeah, I think we do a lot of we develop a lot of unique technology to clinical use. However, for example, we don't know exactly the mechanism. For example, interferon alpha, we know there's effective for enhance the graft versus leukemia effect, but we don't know the mechanism. We don't know that which molecular or which pathway the interferon alpha to have the activation. So because we don't know the mechanism, so and also interferon alpha has a lot of side effect. So I think maybe we can with some kind of cooperation with the college in Michigan, because we know the college in Chicago also use interferon. That's what I think. But how to develop the protocol, I think we need to discuss.
Okay. Very good.
Actually, we have a question online by the noise, and the question is, I think it's probably for Wei Ping. I don't know. It says that nowadays it seems that studies in immunology or tumor microenvironment adopt many fancy molecular and genetic tools. Is there a place for morphology, say pathology, in these studies? If yes, where is it?
I try to say something in this case, not necessarily I have an answer. It's a great question, I have to say. So everybody knows we start, when you look at the molecular pathways and the genetic pathways and different tools, and we have to keep in mind that the cells has its morphology, its shape. So that's very important. Actually, when you see different types of cells, particularly cancer cells, the morphology is first of all, the morphology are so different. And then the invasiveness is different. So this depends on where the cells are located and how can they become more aggressive and more invasive. It's very obvious the morphology is the key. So the thing is, how can we combine the molecular and genetic information with the morphological information? This is a key question, you know, and I don't think anyone has any way, a good way to go, you know, because when you culture the cells, you basically change the morphology. So that's the pitfall actually. And we need to know the information. Of course, these concerns about physics, for example, the stiffness of the cells, the skeleton, and those things we need to work together. We have a little work in that space. We start to work on how the mechanical signal is related to biochemical signals, and how the stiffness of the membrane, the structure of the cells will be affected. You know, I just only have this basic impression here, and we could talk more about this, but absolutely great question.
Well, thank you. And I'd like to thank all of the speakers today, and also everyone who's participated and asked really good questions. And Joe wants to make a few concluding remarks.
Well, first of all, what an incredibly high quality conversation. And so I'm very appreciative to Max and to Ning, but also for their interest in trying to lead this group forward. The null hypothesis for you researchers is we don't need another structure to do inquiry. You're all succeeding without each other. You know, you can share at conferences in the journal. We don't need anything more. But what we've seen in the JI is actually when we facilitate collaboration and remove some of the barriers that are sometimes regulatory within our own countries, getting people together to explore, we can actually have a different level of creativity than you can do on your own. We've been able to identify resources here at Michigan with matched resources there at Peking University to really prioritize cancer. And you're just starting to tap on, for somebody who's not deep in the field, really exciting possibilities. In addition to leaving you with my thanks for this conversation, if I could just leave you with the challenge, how can you start to explore things that you could do together that would be really synergistic, that would advance the field, but also show how we can do things together in ways that I think benefit both of our own countries. I think the world is looking for examples on how we really co-create together, and we know we do that, but I think we have a chance to really shine the light on this kind of behavior in a positive way for the world to see. So I'll be extremely interested in facilitating, enabling Ning and Max and all of you, is we find really creative opportunities to showcase cancer in the JI. I know people have places to go, and so I will sign off now. A special thanks to Dr. Amy Huang and the team behind the scenes for lacing this all together. Unbelievable turnout. You know, I saw the numbers were up to 96 at one point. And this is at a time period that's either before the start of the day here at Michigan or after the day is over there in Beijing. So there's interest and enthusiasm, and thanks for sharing that with me. Have a good evening, Beijing, a good day, Ann Arbor. Happy holidays, everyone. And we'll see you next time.
Thank you, Joe. Thank you.
Thank you, Joe. Thank you, everyone.
Okay.